So, in our last discussion, we saw that the force of natural selection goes down as an organism becomes older and older, and this happens because the contribution towards overall reproductive success is much greater from the earlier age than towards the later age. And that is why there is this concept of selection shadow, which says that somewhere during the later part of life, if there is a mutation which ends up having a detrimental effect on the reproductive success, then the overall effect of that mutation on the fitness is going to be very less. In other words, the ability of natural selection to take out a detrimental mutation towards later part of it, that expresses towards the later part of its life, is much less compared to the same mutation if it expresses towards the earlier part of the life. So, what does the data tell us? That is what we are going to discuss during this particular session. Now, the evolutionary theory is suggesting that the fundamental cause of aging is the fact that there is non-zero mortality during every phase of life. So, if that be the case, then that automatically leads to the prediction that if there is greater extrinsic mortality in a population, then that population should age faster and have a lower lifespan. Why is that so? Because if most of the individuals are dying early in life, then all the mutations which are going to express only later in their life, those mutations are typically not even getting expressed. The carriers are dying even before those mutations play a role, and therefore, in this way they will end up escaping selection. And when they escape selection like this, now if you bring them under a benign condition and if you let the organisms live as long as they want to, then these mutations will get a chance to express and their deleterious effect will show up as aging or show up as death at an earlier life. So, that is why we expect that high levels of extrinsic mortality is going to lead to faster aging. Now, do we have any reason to believe this? Turns out that if you simply look at the data, there are quite a few evidences which suggest that this is really the case. So, for example, flying. Okay. We know that if an organism is able to fly, then the number of organisms that can predate on that organism are much less. In other words, flying reduces predation rates, and predation is one of the major sources of external mortality. Therefore, we expect that flying organisms which they should in general live longer. Now, turns out that this is borne out in multiple cases. So, if you look at birds, they typically live longer than similarly sized non-flying mammals. Similarly, among mammals, the flying mammals are the bats, they typically end up living much longer than all other mammals. Similarly, if you look at insects, insects typically have a high external source of mortality and therefore, they generally live short. The very nice exception to this rule are the queens of social insects like ants and termites, who actually have a very low source of mortality because they are well protected by everybody else in the nest. They also get lots of food that is provided for by the other people, and therefore, their rate of external mortality is low, and therefore, we normally see that their longevity is very high. So, this is very much in line with what we said, that external mortality should lead to high levels of external mortality should lead to faster aging. Do we have more concrete evidence? And more importantly, what we just now looked at was across species, right. So, across species, so many other factors can be at operation. Do we have something under controlled condition or under known condition within a species? And turns out that we do. So, this is one of the very famous examples. Before going to the example, let us look at what the prediction is. So, the prediction is that if there is reduced ecological mortality, that is external mortality, then there is going to be increased selection on late acting deleterious mutations. Why? That is because the organisms will be able to live longer, and therefore, all those mutations will be under selection, and therefore, they will age slower. On the other hand, if you have increased external ecological mortality, therefore, most of the organisms will die young and the selection on late acting mutations is going to be much less, which basically means organisms will end up aging faster. Okay. So, here is a pretty famous example of this. So, this is a study on the Virginia opossums, commonly simply called opossums. These are marsupials and they are found in the North American continent. So, this organism has this peculiar tendency that if it feels really threatened, then it actually plays dead. Okay. And this allows it to escape many predators, particularly those predators which do not typically touch dead animals. Very interestingly, from a conservation point of view, this actually has played a negative role in their life, because what happens is, suppose a lot of stress that they get is from vehicles on roads. So, basically when they see a vehicle approaching at a fast pace, they simply play dead and therefore, they end up becoming part of roadkills along highways. Anyway, so this particular study was done by Ostead, Stephen Ostead, a big name in the field of gerontology, and here he used two populations of the Virginia opossums, one that were found on a mainland and one that were found on an island called the Sapelo Island, somewhere close by. So, turns out that on the mainland there were lots of predators and therefore, there was a much higher rate of predation. On the island the rate of predation was much lower. But apart from this difference in predation, these two sites, they were actually extremely similar to each other, and Ostead actually spends a lot of the paper to show that temperature, precipitation, external parasite load, availability of resources, etc., everything is as similar as possible between these two sites and not significantly different. Great. So, for this study, what Ostead does is he radio collared a large number of females shortly after their birth and followed them throughout their lives and essentially recorded when they died. And he had also recorded other kinds of data that I am going to talk about. Now, what was happening in these two islands because of the differences in the predation rate? So, turns out that here we are showing you the survival proportion surviving on y axis and the age of the females on the x axis. Now, note that here this proportion surviving, we are putting it in natural logarithmic scale, okay, because of which this as I showed you, typically this falls negative exponentially, survivorship in normal arithmetic scale falls negative exponential, but if you do a ln transform, then they are going to fall linearly, and this that is why these values -1, -2, -3, this is in survivorship is going to be in decimals, right. So, that is why when you do a log transformation, they come out in negative number, in case you are wondering why we have negative number on the y axis. Now, you can clearly see that on the mainland the survivorship is much lower corresponding to the island. So, for example, if you look at this around age 30 months or so, almost all the individuals are dead on the mainland, whereas quite a large fraction of them are still alive on the island, right. So, if you look at the values in terms of longevity, the mean longevity on the mainland was about 20 months, on the island it was about 24.6 months. More importantly, the maximum lifespan found was 31 months on mainland, on the island it was 45 months. So, these are all statistically significant differences. So, this clearly shows that there is a huge difference between the survivorship on the mainland and the island. So, now what is my prediction? Based on what I just told you in the previous slide, our prediction is that on the island, because the ecological mortality is low, therefore, there will be increased selection on late acting deleterious mutations, which means that the island populations are expected to age slower, right. Was that really the case? So, what Ostead did was he looked at two parameters to figure out the rate of aging. One parameter, both are physiological parameters. One parameter was in terms of the growth rate of the litters. So, turns out that these opossums, they give birth to their young ones over multiple years. Now, when you look at the rate at which the litter grows in the first year versus the second year, for the mainland populations, the litter growth is much slower in the second year compared to the first year. On the other hand, if you look at the island population, there is no difference. In both years, they are essentially growing in the same rate, the litter is growing in the same rate. So, this is suggesting that for the mainland population, the ability of the females to provide nourishment for their kids, that ability is going down from first year to second year. Now, you could have said that, hey, what if this is because of difference in availability of food? But remember, I told you Ostertag had explicitly shown that there is no difference in the amount of food that is available, that is same across both the places. So, this clearly suggests that this is related to how much the mother is able to provision for the babies, and that ability is going down for the mainland population, not for the island population, suggesting that the mainland population is probably losing its physiological functioning faster than the island population. Similarly, the other thing that Ostertag looked at was the brittleness of the connective tissues. Now, you know that when an organism, for example even humans, when we are young, our connective tissues are very flexible. Now, as we grow older, certain connective tissues, collagen in this particular case, they become much more brittle. Why does that happen? That happens because there are cross linkages in the polymer that start taking place, because of which aged organisms, their connective tissues become less flexible and they tend to break faster. And what they ended up showing, Ostertag ended up showing, was that if you look at the rate at which the tissue was becoming brittle, this was happening much faster, significantly faster in the mainland populations as compared to the island population. This again is an indicator that the island population is aging slower. So, this was an experiment where a field experiment to show that ecological mortality can affect aging. A similar experiment was done in Drosophila populations. This is Stearns et al. PNAS 2000, where they took two sets of populations, three populations each. In one population, they externally imposed high levels of adult mortality, they call it the ham population, high adult mortality, and there was another set of populations where they did not expose them to high level of mortality, they call it the low adult mortality lamb population. So, if this logic that I am showing you over here is correct, then the ham populations were expected to evolve faster aging, and that is exactly what happened. They ended up showing that the ham populations were aging faster and their mean longevity also went down. So, this is another direct test where people showed that evolution, because of high adult mortality early in life, can lead to faster aging. So, now the question is that how exactly will a selection shadow end up affecting aging? And turns out that two major mechanisms have been proposed. One is known as mutation accumulation, the other is known as antagonistic pleiotropy. The two are actually very close to each other, and I will quickly show you what they imply through our Excel demonstration, the one that we had used earlier. So, this is what we had done, right? And so, if you remember this simulation, we showed that the lifetime reproductive success is this value 19.80 in a population in which there is no aging and the density independent mortality rate remains constant at 0.1 and the reproduction at every age class remains constant at 2 individuals. So, this is the sum of this, and what I will do is I will just copy paste it over here for reference. Okay. So, this is the reference value. This is when there is I will just call it the reference value, that is all, right. So, this value 19.806 is this value, the sum of reproductive success at all the age classes. Now, suppose you have a mutation which only expresses its bad effects late in life. Okay. So, what kind of bad effect? Let us assume for a moment that this is something that ends up reducing, let us say, the fecundity in later life. Okay. So, let us assume we have this, and let us say 1, 2, 3, 4, 5. So, let us say for these 5 cases this fecundity basically becomes 0. Okay. So, okay, let us start from here. So, 0, 0, 0, 0, 0, right. So, here is a mutation that has ended up causing the individual, the organism, to lose all reproduction for the last 5 age classes, and you can see that the overall effect on fitness, it has gone from 19.8 to 19.67. Okay. So, this is what I meant by saying that if there are late acting mutations, then the effect of that mutation on the overall reproductive success is going to be very less, and therefore, selection's ability to weed out this mutation is going to be very low. So, mutation accumulation theory simply says that deleterious mutations which express only late in life, they will tend to accumulate in the population, and when that happens, these are going to show up, these bad effects are going to show up as aging. So, this I showed you what happens if you have let us say reproduction problem. Okay. So, now let us make it even better. We said that reproduction is contingent upon survivorship. So, now let us have a scenario where this thing, this mortality becomes 1. Mortality becoming 1, as you can see, basically means that all individuals are dying, right. So, I have made it mortality equal to 1 everywhere, and you can see that even if I have made it 1 for 5 cases, from 19.8 it has gone to only to 19.63, right. Again, saying that it does not matter whether it affects survivorship or reproduction, late acting mutations are typically going to have very little effect on fitness, and therefore, they will tend to accumulate in the population. So, that is the mutation accumulation stuff. Let us make this again 0.1. Okay. The other thing is what is known as antagonistic pleiotropy. What is antagonistic pleiotropy? So, we know when one mutation or one allele ends up affecting multiple traits, that is what is known as pleiotropy. Antagonistic pleiotropy is a special case of pleiotropy where a mutation has positive effects early in life, but negative effects later in life. So, let us see what will happen in such a scenario. So, let us assume that we have a mutation which increases reproduction in the first age class by, let us say, 3 units, from 2 to 3 units, but at the same time it leads to death, let us say for let us say it makes reproduction in the last 5 or 6 age classes to 0. Okay. So, something like something like this, correct. Look at what has happened. This one reproduction has gone up by 1 unit, 5 reproduction in 5 or 6 age classes have been completely lost, actually 7 age classes have been completely lost, yet overall the fitness has increased, the lifetime fitness has gone up, right. It is clear why this is happening, right. I said that any change that happens earlier in life has a much greater effect on the overall fitness than any change that happens later in life. So, here a small change here, large change here, but overall the effect is positive. In other words, such a mutation is going to be selected for. Now, if you have a scenario like this, what this is saying is all mutations which have beneficial effects early in life are going to be favored even if they have bad effects later in life, as long as the overall effect on the fitness is positive. Okay, a case like this. So, such scenarios are known as antagonistic pleiotropic scenarios. So, with this we will go back to our slideshow. So, here are the definitions. So, the mutation accumulation theory is typically okay. The idea for this comes from the work of Ronald Fisher and Halden, but the person who really makes it popular is Peter Medawar in 1952. So, basically the theory is that late expressing mutations will accumulate more readily in the population as the force of natural selection declines later in life. If these mutations are deleterious, their deleterious effects will show up as aging. Antagonistic pleiotropic theory, actually the roots of this theory are also in the works of Peter Medawar, but the person who makes it really popular and kind of shows it in more detail is G.C. Williams in a 1957 paper. And what he says is that genes or mutations can have impact at multiple ages. All those mutations which increase early fitness can be selected even if they negatively affect late life fitness if the overall reproductive success, as I showed you, is positive. And therefore, he suggests that there is always a trade off between early fertility and later fertility or survival. In other words, anything that increases your reproductive fitness early on in general will be selected even if it has some negative effects later on in life. So, just to compare these two things, under the mutation accumulation theory, there will not be any difference early in life, but late life the performance, the fitness will go down. Under the antagonistic pleiotropy theory, the performance in early life will be favored, will go up, but the performance in later life is going to go down. Now, in practice, it is actually very difficult to differentiate between these two hypotheses, and frankly speaking, they are not really mutually exclusive either. Okay, something these two kinds of mutations or these two kinds of effects can happen in the context of different traits. So, we will talk about some empirical supports both for mutation accumulation and for the antagonistic pleiotropy thing. So, this is a selection experiment in which people took housefly populations and made them to reproduce early in life, that is 4 to 5 days after eclosion. Okay. So, if you are making them reproduce earlier and earlier in life, then you expect what? All the late acting mutations, those mutations are going to go out of the purview of selection. In other words, aging is going to happen faster or their lifespan is going to go down. So, what they did in this particular case is that this should reduce the selection on late acting mutation leading to increased senescence. So, what they did was they did the selection and every few generations they actually allowed the longevity to play out the entire lifespan, and then they saw how the maximum lifespan was looking like. And as you can see, within about 25 generations, both in males and in females, the mean longevity in days went down, and it went down quite a bit, from about 37 to about 28 or so, about 9-day reduction in females, and from about 28, 29 to about 25, about 4 to 5 days reduction in the males. That is a pretty large reduction. Interestingly, although I am talking about this in the context of mutation accumulation, if you think about it closely, you will realize that antagonistic pleiotropy can also lead to a similar effect. So, that is why I said typically it is very difficult to distinguish between the mutation accumulation and the antagonistic pleiotropy experimentally. What kind of support do we have for antagonistic pleiotropy theory? Remember, it says that anything that is going to have a positive effect or positive value early on in life will have a negative value later in life. That is antagonistic pleiotropy. So, examples were from this bird called collared flycatchers. So, here there are certain birds which start breeding at the age of 1, whereas some birds they do not breed in the first year of their life, they start breeding in the second year of their life. So, Gustafsson and Part, what they ended up showing was that if you start breeding earlier in life, then your clutch size although it goes up, it does not go up too much. Okay. Whereas, if you end up foregoing the reproduction in the first year of life, okay, no reproduction there, then your clutch size starts much higher and actually goes up much higher and stays much higher for your entire life. In other words, the early breeders are paying a cost by having a lower clutch size. Now, very interestingly, if you do the kind of analysis that we did for the survivorship multiplied by clutch size or mx. So, clutch size is basically mx in our simulation. So, if you multiply their year to year survivorship by that mx, and using this you compute the lifetime fecundity, the lifetime reproductive success, you find that despite having a lower value of mx throughout their life, it is the early breeders who actually end up having a greater lifetime reproductive success. Okay. But the main point we are trying to show over here is that if you start breeding earlier in life, you pay a cost by not being able to increase your clutch size. Similarly, you remember when we were talking about the experiments on Drosophila for enhanced lifespan, there was another experiment that I talked about, which was that of Michael Rose in 1984, which was reported in 1984. So, this is a very famous line of work where they had two sets of populations, 5 populations each, B populations called the baseline populations, these were the controls, and the O populations called old populations, these are the ones which were selected for increased lifespan. So, basically every few generations they would collect the eggs later and later in life. So, in order for a population to be able to reproduce, they had to first survive to that age and then alone could they reproduce. So, this way there was a selection for increased lifespan, and there was a massive response. The O populations had a much greater mean lifespan compared to the B populations, but more importantly what was shown was that, the prediction over here was that there will be a trade off between early life and late life fecundity. In other words, the O populations will be able to increase their lifespan only by sacrificing their early life fecundity. That was the antagonistic pleiotropic thing that we are talking about, and that is pretty much what ended up happening. So, if you look at this, this is the age on x axis, and what you have on y axis is the net difference between the O and the B mean fecundity. So, mean fecundity of O minus mean fecundity of B. And what you can see is that the early part of the life, this is actually negative, which basically means B's have greater fecundity than the O's, but in the later part of life it flips. Now, the O's have greater fecundity than the B's, which essentially means that the B populations, they are having an antagonistic pleiotropy, they are increasing their fecundity early in life at the cost of reduced late life fecundity. And the O population, they are increasing their longevity, but for doing that the early life fecundity that they are having, that is going down, and because of that all those genes which would have made them better in early life but worse in late life, those are being selected against. So, this is consistent with the antagonistic pleiotropy hypothesis. These two are not the only examples. This particular thing that long lived populations will have a trade off between early life and late life fecundity, this has been seen again and again. So, for example, if you remember, we talked about the Luckinbill et al. Experiment of 1984. They had also selected for longer lived Drosophila populations. They also reported a reduction in early life fecundity. Then there was a 2015 review by Lemaitre et al. And they ended up showing that these guys were looking at natural populations, 12 bird populations, 10 mammal populations and 2 reptile populations, and out of these 26 cases, 21 cases ended up reporting a trade off between performance in early life versus late life. In other words, those organisms which were living longer, they had a dip in their early life performance, but an increase in their late life performance and vice versa. So, to conclude, organisms, according to evolutionary theories, organisms age because selection largely overlooks those organisms who live to late life. and or because selection has favored individuals that seek early benefits even at the expense of later costs. So, the first part, accumulation of mutations, that is the mutation accumulation theory, and the second part, selection has favored early benefit even at the expense of later cost, that is the antagonistic pleiotropy theory. Here I must mention a variant of the antagonistic pleiotropy theory, also known as the disposable soma theory. So, antagonistic pleiotropy says that if there is a mutation which is having positive effect early in life, then effect, even if it has negative effects later in life, it is going to be selected. Disposable soma theory also talks about a trade-off, but it talks about a trade-off between reproduction and long-term somatic maintenance. So, basically this theory says that, look, for the fitness of an organism, it is the reproduction which is more important, and once the reproduction has happened, then even if the organism is not really maintaining or putting much resources towards somatic maintenance later in life, that does not really affect its fitness too much. Therefore, any mutation which will end up benefiting the reproductive output earlier, particularly earlier in life, even if it has negative consequences for the somatic maintenance later on, it is still going to be selected. And this negative consequence for later life fitness is what shows up as aging. So, the disposable soma theory is also a well known theory, but since it is essentially talking about a trade off, many people have argued that this is nothing but a subset of the antagonistic pleiotropy theory, right. This looks pretty clean, and as I showed you, lots and lots of empirical evidence as well, but this leaves one nagging thing, one nagging doubt. What is it? That is in the context of immortal organisms, right. There are certain organisms who we apparently call as immortals. What about those? If this evolutionary theory is correct, then we should have no immortal organisms, right. So, where are immortal organisms coming from? So, I will just deal with one case study over here. Perhaps the first thing that will come into anybody's mind when they talk about immortal organisms, and that is this organism called Hydra. So, those of you who, just in case you do not know what a Hydra is, it is a very small tiny organism, it is a Coelenterata, it is a very early evolving taxa, and turns out that press and all popular science, they just go bonkers about this particular organisms, because this is the name they gave it, the immortal Hydra. Now, the first thing you have to realize is that there is a slight bit of a catch over here. When they talk about it in the popular press, when they use the word immortal, they seem to mean that this organism cannot die. That obviously cannot be correct, right, because if that were indeed to be the case, the world would have been full of Hydra by now, and obviously that is not happening. So, turns out that this is not immortal in the common sense of the word, because they can and they do die due to all kinds of external reasons. It is not that they cannot die. Hydra is immortal in the technical evolutionary sense of the word, because essentially what happens in them is that they have these stem cells which can renew themselves indefinitely. So, even stem cells, remember, they cannot keep on dividing forever. Most stem cells will stop dividing at some point, right. But the Hydra stem cells are special in the sense that they can keep on dividing indefinitely, and they actually show no signs of cellular aging. Now, turns out that Hydra, they reproduce asexually by budding. So, if you look at this particular picture right at the bottom, you see that bulge towards the left, that is essentially a bud. So, this is how most Hydra will reproduce. At some point they will create that bud, and then that bud will just detach from the main organism, and that bud will form a new Hydra. Now, since they can reproduce by budding like this, and since their stem cells will keep on dividing forever, therefore, they can keep on budding indefinitely, and it is in this sense that they have been called immortal, okay, that they can keep on dividing forever, not because they cannot die. So, this needs to be kept in mind. Now, obviously the question is, what is happening over here? Why is it that the stem cells of Hydra can divide indefinitely? Now, this is a question that people have really looked at in great amount of detail, and among other things, one of the major properties or one of the major regulators of this thing has been this gene called FOXO. So, turns out that if you knock down this gene, as this was done by this paper Boehm et al., then this property of continuous division actually gets arrested, right. Now, this is a bit exciting because FOXO is a very important gene that actually can be found across pretty much all organisms that is there. I mean, up to human beings, everybody has them. So, looks like FOXO might be one of the keys. It is certainly not the only key, it might be one of the keys for aging. However, I mean, when you try to knock down or you try to impact FOXO across many taxa, you see that aging gets affected, but that does not mean that FOXO is the only thing that affects aging, right. Similarly, you have insulin and insulin related pathways. They are again very critical components in the metabolic pathways of many organisms, and again they seem to be playing a big role in aging, but are they the only ones doing so? Absolutely not. So, there is some promise that one day we will be able to find some common genes which might end up affecting aging in multiple scenarios, but it is also clear that they will not be the only ones that will determine aging in that particular organism. They will be, their effect will be regulated by many other things. So, again I want to emphasize this very critically, that although I talked about the evolutionary theories of aging and the mechanistic theories of aging, these two theories are actually operating at two different levels, proximate versus the ultimate, and therefore one is saying why aging happens at all. The other is saying, the mechanistic theories, they are saying that for a given organism, what are the factors or what are the molecules that are causing the aging in that scenario under those whatever environmental factors. So, a proper study of aging requires you to take both the evolutionary and the mechanistic aspects into account. Without that, not much will happen. Also appreciate, and I told you, right, 3 lakh papers were published in just 10 years, and many lakhs were published before that, and probably a few more lakhs have been published between 23 and 25. So, it is impossible for me to give you a very good and in-depth treatment within just a few lectures, but what I have done over here is to just give you an overview of the various lines of thinking about why organisms age, but then there is a lot more that is there to it. There are a lot more information, and maybe some of the papers that I have cited during these few discussions, they will allow you to look into those details. So, we end our discussion of aging over here, and the next pattern that we are going to talk about will be sex and sexual selection, but that is what is going to happen next week. See you then.